screen now. I think you can all see it. Um, yeah, thank you so much for inviting me. I'm really, really happy to be here tonight. This is also very exciting for me because this is actually my last night of COVID isolation. So the fact that I'm like interacting with people outside of my family, super, super exciting. I'm very anxious to leave my attic where I've been for, it feels like a long time. Um, but yeah, as Bronwyn mentioned, I'm an associate professor of anthropology at the University of Cincinnati. And I've spent really the bulk of my career, or the bulk of the research over the course of my career has really focused on understanding variation in human pigmentation, as well as sort of the evolutionary factors that have shaped that variation. And so as you can see here in this series of images that are taken from the artist Angela Daas's project Humanity, humans show a really, really remarkable array of variation in both skin, hair, and eye color. And as I kind of said before, you're all experts in humans, so you don't really need this art project to know that, but I think this is a nice visual representation of that. And so as a student of anthropology, and in particular as a biological anthropologist, I've always been really intrigued by human phenotypic variation, its genetic basis, and as I said, the evolutionary forces that have shaped that variation. And if you're into those types of questions, I think pigmentation is a really great trait to study. So what I'm going to do tonight, I'm going to kind of focus on a few areas. So first, I'm going to talk a little bit about what causes variation in pigmentation, right? The actual biological factors that contribute to it. And I'm also going to talk about how we measure that as scientists, right? So Bronwyn, your thought for a breakout question was like, how many categories of skin color are there? We're going to toss that out the window because when we think about skin color, we actually think about it in a different way. So I'll talk about how we measure it and actually what the extent of that variation looks like across our species. And then what I'm going to do is I'm going to walk you through some of the predominant hypotheses for how all of that variation came to be. Okay, so working within an evolutionary framework. And then I'll wrap that up by talking about some of the evidence we have that supports or doesn't support those hypotheses. And I'm what's known as a molecular anthropologist, which means that I use genetic and genomic data to investigate questions relating to human variation and evolution. So we're going to talk about genes, right? Which is where my icebreaker question came from. Okay. So let's get started. So first, just a little bit of history, right? We're pretty confident that humans have varied in pigmentation for a very, very long time. And it's hard for us to know exactly how long and the extent of that variation, right? But if we wanted, we could go back and try to draw some inspirations from different artworks, right? So for example, one of the places we see this is in this banquet scene. This is from a fresco at the Tomb of the Leopards in Tarquinian, dated to about 480 to 450 years before Common Era. We can see these people are depicted with varying skin tones. We also see the same thing in the mural below from the Mayan city of Bonampak, dated to about 1200 years ago. Okay, so, these artistic representations can show us, hey, yeah, people in the past noticed this and they paid attention to it enough, at least to record it. They don't always tell us so much about what people in the past maybe thought about this variation, if they had any ideas about how it came to be, if it had any specific meaning to it. Sometimes we can turn to ancient writings. So for example, we know from the writings of Herodotus, the Greeks kind of thought that variation in skin color was probably somehow tied to geographic origin, right? And they had some ideas that it was shaped by climate. Specifically thinking like hot places make you look different than cold places. So we know these are questions people were thinking about. Now, if we kind of jump forward in time and we take a very kind of a very narrow sort of Western scientific perspective, right, we start to see skin color being used as a key designator of different groups of humans, right? So if we go and we look at Carolus Linnaeus's Systema Naturae, right? Linnaeus identifies 4 groups of humans. Okay, actually, he identified 5, but one group was monsters, and we're not going to talk about monsters today. But the 4 groups that he identified, right? He identified these 4 groups. He divided them by geography, right? So we've got humans from Europe, humans from the Americas, humans from Asia, humans from Africa, and and he identified a key defining feature of all of those groups, which was skin color. Okay. Now, Linnaeus also did some other things when he was defining those groups, right, that go beyond just scientific observation, right? He assigned some other characteristics to them, things like sort of uniform clothing styles and styles of government and temperament, things that become a lot less scientific and that then also lend themselves to the use of these categories, and particularly the way they're tied to skin color, to create a sort of a hierarchical ranking of human groups. Now, anthropologists and human biologists today know that this isn't really a valid way to sort people, but the impact of that, right, and what he was doing in making these divisions fits in very well with what we know is going on in the socio-political climate of the time, right, where we've got colonization and enslavement. These categories feed very nicely into that, right, and it gives sort of a scientific what's the word I'm looking for? I'm forgetting the word, but it's lending scientific credibility to that. Nowadays, scientists would not support that, but I bring that, and that's really that's the topic of a different talk. But I bring this up because as someone who studied skin color for 20 years, I think it's really important, as fascinating as I find this as a biological trait, to think about the history of how it's been used and misused in our past. Okay, so stepping out of history, let's jump to the now and talk about what we know about pigmentation biology. Okay, so variation in pigmentation that we see in our skin, hair, and eye color is largely due to melanin. So melanin is a complex biopolymer, and it's produced in specialized cells known as melanocytes. Okay, melanocytes are found in the basal layer of our epidermis, and that's kind of what this pink blobby looking cell is here. Okay, within melanocytes, melanin gets produced and packaged in specialized organelles known as melanosomes. Okay, and those melanosomes, when they mature, they migrate up through those dendritic arms of the melanocyte, and they get transferred to surrounding keratinocyte cells, and then those cells sort of migrate up through the layers of your skin. Now, when we're thinking about causes of variation in skin color, we might think, oh, well, maybe people with darker skin just have more melanocytes, right? That seems like that might make sense. In fact, that's not the case, right? So melanocyte count from person to person is pretty constant, okay? It might vary in an individual across body sites, but in general, from person to person, regardless of your constitutive skin color, melanocyte count is the same. What's really important when we're thinking about differences in skin color is the amount of melanin that's being produced in those melanocytes and the type of melanin. Okay, so melanin actually comes in 2 different flavors, if you will. There's a brown-black eumelanin, which actually has 2 different subtypes, but for tonight, just one. Just think of brown-black eumelanin. And the other flavor of melanin is red-yellow pheomelanin. As you can see in sort of this cartoony diagram of the melanin synthesis pathway, the initial steps in the production of both eumelanin and pheomelanin are the same. It's not till the end that pathway sort of diverges. Now, how does this affect pigmentation? Okay, so I told you that what we really want to pay attention to is the total amount of melanin you're producing, as well. And the other thing we want to pay attention to is the ratio of pheomelanin to eumelanin. Okay, so someone like me, right, you can see my image on the screen. In general, my skin is not making a ton of melanin either way. But when you look at something like hair color, for example, okay, I don't know if you can see with the lighting, but I have this reddish orange hair. My hair has a very high pheomelanin to eumelanin ratio. Okay, I'm making lots of that red-yellow pheomelanin up there, not so much of the brown-black pheomelanin, or brown-black eumelanin, sorry. Whereas someone with darker hair, it's going to be the opposite. They're going to have a lower pheomelanin to eumelanin ratio. Okay, so again, when we're thinking about variation in skin color, we're thinking about total melanin produced, okay, as well as the ratio of these 2 types. Okay, great. So we know pigmentation is affected by melanin. How do we like measure or assess this in people? Because it turns out melanin is actually a tricky compound to work with, right? And so first of all, um, if you had biological tissue to isolate it from, that's kind of a complex, messy chemical process. It's a pain to do. Most people don't do it. Also, as someone who has spent a lot of her career taking samples from people, I don't want to take skin samples from my subjects, nor do they want me taking skin samples from them, right? We want to be as non-invasive as possible. So in the past, right, and so past, think mostly like pre-20th century, one of the objective ways that people would try to assess skin color was by using some sort of standardized set of colored, in this case, these are color tiles. This is a set of color tiles known as the Van Luschen tiles. And the idea is you would sort of hold a person's skin up against those tiles and try to find the best match. Okay. So the thinking is if the tiles are standardized, this should be pretty objective. It often was not. Okay, there could be a lot of inter-observer disagreement. Also, people realized, oh, depending on the lighting, matches to a particular tile color might be, might vary. The tiles get scratched, they get dirty, they get dusty. This really wasn't a great system. And so then right around the 1950s, maybe a little bit later, a new method was developed that was going to be a lot more objective and hopefully a lot more precise. And that's when we start to see the development of devices known as skin reflectometers. And these basically work on the principle that you have an instrument, it has a probe with a light attached to it, and you would basically hold that probe up against someone's skin and shine the light on the skin, and the device would measure how much light is reflected back, right? And we know that darker things are going to reflect relatively little light, really bright things or light things are going to reflect a lot of light. Okay, and so after that, we see sort of this boom in anthropometric research where there's all sorts of skin color studies happening using these skin reflectometers. But these were also kind of tricky to work with, okay? They were portable, you could take them into the field, but they were a pain in the butt to work with. So for example, as I mentioned, you shine that light on the surface of your skin, but what you were actually supposed to do to use these devices correctly was you were supposed to pass that light through a series of 6 or 9 filters, colored filters of known wavelength, depending on the maker of the machine, right, that you were using, and record your measurements that way. And I don't know if any of you have ever been part of a research study where scientists were taking measurements. I assure you, absolutely no one wants to sit there while someone like me changes a filter 6 to 9 times and takes that many measurements at all different sites on your body. So what happened in practice was most scientists didn't use all those filters. They might use 1, they might use 3. Maybe the filters would break, right? Because they're made of glass. If you're on a 6-month field trip and your filters break in month 2, you're kind of out of luck. So we had a lot of data being collected, but it wasn't always collected in the same way, which sometimes made comparing across studies kind of challenging. But then in the 1980s, a new method was developed, still based on skin reflectance, but it said, hey, let's actually focus on measuring reflectance at the 2 primary chromophores of the skin, the 2 things that matter, right? We've already talked about one of those chromophores, it's melanin. The other chromophore is hemoglobin, right, which is found in your blood. And so it turns out that melanin and hemoglobin have some pretty distinct absorbance profiles. So this chart is just showing you we've got wavelength on the x-axis, the y-axis is absorption. And again, this is sort of an idealized graph just to kind of show you what's going on. But one of the things that happens is, although melanin, right, which is shown by this dashed line here, has sort of this kind of steady gradual decline in absorbance as we move across the spectrum, hemoglobin is a little bit different. We can see it has these peaks of absorbance, and then it has a very sharp dip in absorbance right around these red wavelengths, okay, which means it's reflecting a lot of red, which is one of the reasons why we perceive blood as being red. And so what these devices, which are known as narrowband reflectometers, do is they shine a red light and a green light on the skin at very particular wavelengths associated with sort of maximal differences in absorbance of hemoglobin and melanin. They measure how much light is reflected back, and one of the outputs that it can give you is an estimate of how much light is reflected back off of the skin or the hair that is due to melanin. Okay, which really, as biologists, melanin is what we're trying to measure. Okay, and so that value is something that's known as the melanin index, or M index. And so the way it works is higher melanin indexes indicate darker skin, lower melanin indexes indicate lighter skin. Okay, these devices are really, really easy to use. So the image I'm showing you up top, that's an older model, but here I am using that older model. This is when I was a graduate student 20 years ago, looking at skin color variation in Melanesia. These things run off a 9-volt battery. They're very, very, very easy to use. I love them. More updated models now will measure melanin index, but they'll also measure color in other more complex ways if that's what you want. And so we're seeing a real shift in the discipline to using kind of simple devices like this that allow us to measure. We're not directly measuring melanin, okay, but this is a rapid, non-invasive way to estimate melanin, and it seems to be pretty accurate. Okay, so when we use devices like melanin index, what does that look like in terms of actual human phenotypic variation, right? What are the ranges that we might see? Okay, so this graph is just showing you the distribution of melanin index values for a few different groups that we've sampled in my lab, or that close collaborators have sampled over maybe the past 5 to 10 years. And so again, melanin index is what's plotted on our x-axis here. And one of the things you'll see is that for some of these groups, okay, the range in melanin index is pretty narrow, right? That's kind of a key feature that we see in our East Asian and our European American sample. Whereas for other groups, right, like our sample from South Asia, and in particular our sample of African American individuals, there's a really, really wide range in melanin index values. Okay, the other thing you'll notice is that all of these distributions overlap each other. And so that's one reason why using skin reflectometry measures like melanin index really lends those of us that work in this area to emphasize that pigmentation is more of a continuum than a categorical trait, right? That's another reason why those color tile systems were always sort of not satisfying to use, because there's always someone that's in between. Okay, so the use of reflectometry, but I think especially melanin index, really started to change the way we think about this. We think about this as a more continuous phenotype. Okay, so another way we could look at this using sort of a broader sample of populations is shown here. And so this is an image from a paper by Alicia Martin, where she was looking at pigmentation variation and genetics in populations from Southern Africa. But what Alicia did here, and this was when she was a graduate student at UC Davis. I think she's at the Broad Institute now. But Alicia basically took published data on melanin index from a bunch of different populations throughout the literature. And she put them on the same plot, and she arrayed them in order of absolute latitude. So populations that are closer to the poles are on top, and as we move down, okay, we get closer to populations from the equator, right? And these populations in gray here, these are the groups that I sampled in Melanesia. So it's nice to see them being used in other ways. Why did she array these according to absolute latitude? Well, one of the things she was calling out is that when we think about variation in human pigmentation, right, and the way that variation is not sort of randomly distributed around the world, right? It follows a very particular geographic pattern. And what that pattern is that skin color on average tends to be darker in regions closer to the equator, and it tends to be lighter as we move away, right? And you don't need a million fancy studies of melanin index to tell you this, but the data sort of confirms what we see. If you prefer a different kind of image, this is an image from a review paper by the anthropologist Nina Jablonski that also sort of demonstrates that particular relationship. Okay. And, as I said, this relationship wasn't a secret. We didn't need lots of anthropologists going out and measuring melanin index to tell us this. But when we see a particular trait that we're interested in showing a geographic pattern like this, okay, it makes us ask questions, right? And so one of the things that we might think of as anthropologists and evolutionary biologists when we look at a distribution like this, is we might say, is that trait, in this case pigmentation, is that varying in response to some environmental variable, right? Like latitude is not an environmental variable, it's a proxy for environmental variables, right? And what Nina and her husband, the geographer George Chaplin, did is they said, well, look, the most likely variable that latitude is standing in here for is going to be ultraviolet radiation. So they went to test to see, can we look at published measures of skin color, and can we look at data we have for ultraviolet radiation at different points around the world and see if there's a correlation, okay? And in fact, that's exactly what they found, right? So using published skin color data, as well as some NASA satellite data that was specifically attuned to UV radiation, what they found is that yes, the relationship between skin melanin content, okay, and UV is quite strong, right? So all these regions here that are shaded in pink and purple, this is where UV is the strongest, okay? UV, by the way, also tends to be higher if you're going up in latitude, right? Or up in altitude, sorry, we don't always think about that, but that's the thing that comes out in these studies. But as you move closer to the poles in some of these regions that have this yellow or green shading, UV is weaker. And that is also coincidentally where pigmentation is lighter. Okay, so the work they did was very nice in that it really solidified this relationship that kind of everyone knew about, but like no one had actually explicitly tested. Okay. So we've got this nice relationship between melanin and ultraviolet radiation. What does that tell us about the evolution of all this variation that we're seeing? Okay, so I'm going to go over 3 hypotheses. Okay, all of these hypotheses are predicated on that relationship between melanin and ultraviolet radiation. And more specifically, they all sort of rest on one very key fact. And that is that melanin acts very much like a natural sunscreen. So melanin has the ability to absorb and reflect, in some cases, ultraviolet radiation, preventing, for example, UVA and UVB radiation, which is the most common types that we experience here on Earth, from penetrating deep into the epidermis. Okay, that is a key linchpin of the 3 hypotheses that I'll talk to you about today. And so the first of those is a hypothesis known as the photoprotection hypothesis. And I lead off with this because I feel it's like the gateway drug. This is the one that like intuitively makes sense to everyone. Okay. And so what this hypothesis basically says is it says, hey, we know that exposure to ultraviolet radiation, right, essentially exposure to sunlight, can potentially do a lot of damage, right? In the short term, right, it can lead to kind of painful, but temporarily painful things like sunburn, right? Someone like me has experienced this quite a bit. But we also know that long term, right, if that radiation is penetrating deep into the epidermis and the dermis, it can lead to characteristic mutations in the DNA of our cells, specifically sometimes mutations in the DNA of those melanocyte cells. When that happens, if those mutations cannot be repaired, okay, the end result is skin cancer, right? And there are various types of skin cancer, but melanoma is the deadliest type and the type you want to watch out for. And so what the photoprotection hypothesis basically says is it says, hey, it seems that human populations that are living in parts of the world where UV is really intense, okay, it would be really beneficial living in those places to have skin with a high melanin content. Or in other words, natural selection is going to favor any genetic mutations that tell people, hey, make a lot of melanin. Conversely, if a mutation arises that leads to a production in less melanin, natural selection is probably going to act against that, right? Because having that lighter skin color opens you up to all sorts of DNA damage, right? That could potentially lead to skin cancer. Okay, and so I say this is like the gateway hypothesis because I feel like most of us, especially here in this country, have heard a lot of talk about sun protection, and so this feels intuitively, this makes sense. However, this is probably not the best explainer for what we have going on, and that's solely because when you think about the age of onset for most skin cancers, most of them are happening kind of later in life. Okay, and I know there are exceptions, but typically most people are developing skin cancer when they're adults after they've reproduced. And so what that means is that this idea that melanin has high levels of melanin have evolved as a prevalent trait in these high UVR regions as a way to protect against skin cancer probably doesn't hold up, because having lighter skin in those regions isn't really preventing you from surviving and reproducing, right? And when we're thinking about the evolutionary game, it's not just surviving, you've got to reproduce and pass those genes on to the next generation. So an attractive idea, this is probably doing something, but it's probably not the strongest case out there. Okay, but there may be another reason why having a highly melanized skin is really beneficial in places where UV is intense. Okay, and this falls under what's known as the folic acid hypothesis. Okay, so folic acid, folate, is a B vitamin. If you're pregnant, if you've ever been pregnant, if you know someone who's been pregnant, you've probably heard it said you need to take a prenatal vitamin high in folic acid. And the reason for that, in case you ever wondered, is we know that in pregnant women that have folic acid deficiencies, those women are at a higher risk of having babies with a particular type of birth defect, specifically a neural tube birth defect. And so this is when the spinal column during development fails to close all the way. And depending on when that happens during development, that can be a pretty severe phenotype. Okay, so folic acid has a really, really major effect on human health. Okay, interestingly, some studies have also shown that folic acid deficiencies are associated with meiotic errors of non-disjunction during spermatogenesis. That's a fancy way of saying that when you're making sperm and those cells are dividing, the chromosomes don't always sort appropriately. So a sperm should have 23 chromosomes in it. You may end up with sperm that have 22 or 24 or some other number, right? That is also both of these cases, right? Neural tube birth defects and these chromosomal errors have clear direct effects on reproductive fitness, right? So the argument behind the folic acid hypothesis is that, hey, maybe a highly melanized skin has been favored by natural selection in these high UVR regions as a way to minimize folic acid degradation and protect against these effects. And at the same time, right, this hypothesis would argue any genetic mutation that leads to a big decrease in melanin content, selection would probably act against that, right? Those people would probably be disadvantaged compared to people with darker skin. Okay, this tends to hold up a lot better than the photoprotection idea. Although again, they're based on the same idea, the same principle, right? That melanin is doing something protective in these regions. Okay. So what about places where UV is not so intense? Okay, we can think about parts of Europe. We can think about parts of Northern Asia. UV is not so intense and pigmentation is lighter in these regions. Well, how did that happen? Well, one idea is this could just be what's known as relaxed selection. And so what that means is, if you've got populations living in these low UV places and some mutations arise and start to accumulate that lead to lower levels of melanin, like it's not great, but really it's like, it's no big whoop because the UV in that region isn't strong enough to be doing a lot of damage, right? So it's okay, right? Basically, these mutations are all kind of getting past the filter of natural selection. That's one model. The other idea is that maybe there's actually something beneficial about producing less melanin in these places where UV is weaker, okay? And that's sort of the basis for what's become known as the vitamin D hypothesis. And this is a hypothesis that's been around since the 1930s, right? So long before Nina and George were measuring ultraviolet radiation, and long before people like me were distinctly measuring melanin, this is a very old idea. But so the vitamin D hypothesis, as you might guess, rests on our body's need for vitamin D. Vitamin D is a steroid hormone. It's involved in a bunch of different physiological systems. Probably the most well-known is its involvement in calcium metabolism. So one of the things that vitamin D helps us do is it helps us absorb calcium from our guts and get that distributed all around our body to the places that need it, particularly the skeletal system. Okay, so we know, for example, that people with vitamin D deficiencies may suffer from osteomalacia, which is a softening of the bones. In really severe cases, they may suffer from rickets. Okay, this radiograph is showing an image of someone with rickets. We can see the femora, right? The big thigh bones have bowed outwards. In really, really severe cases, rickets can also lead to a twisting and a deformation of the pelvis. If you are a reproductive age female, that's bad news. Okay, you don't ever want to hear that phrase. So vitamin D deficiencies definitely impact skeletal health, but vitamin D is also involved in other systems, right? It's involved in cell growth and differentiation, and it's also involved in immune response. Okay, great. Hopefully I've convinced you in 30 seconds that vitamin D is important, just like folic acid is important. What the heck does this have to do with skin color and sunlight? Well, you can get vitamin D in one of two ways. So one way is through your diet, okay? And here in the US, we fortify a lot of our foods with vitamin D, but there are foods that are naturally occurring in vitamin D, right? And the most common ones are certain types of fatty fish, as well as organ meats. If fatty fish and organ meats are not on your menu, that is okay. Guess what? You yourself are a vitamin D making machine. So every time you go outside into the sunlight, presuming that sunlight is strong enough and you are out in it for long enough, your body actually starts to make vitamin D. So that exposure to sunlight, when that UV penetrates your skin, it jumpstarts a series of biochemical reactions that ultimately culminate in your liver and kidneys that lead to the production of vitamin D. So when you go outside tomorrow and take a walk at lunch, or you cannot deal with that problem at work and you say, I'm going to take a walk, it's okay. You're making vitamin D. You're taking care of yourself. So now we start to see the connection with skin color, because the way the vitamin D hypothesis works is it says, all right, as anatomically modern humans start moving into lower UVR, moving out of Africa and into lower UVR regions like Africa, parts of Asia, maybe a highly melanized skin that was so beneficial in places where UV was really strong is actually not so beneficial, because just like that melanin is blocking the UV from penetrating your skin and destroying folate, it's now preventing you from making vitamin D. And so the hypothesis says, in these particular low UV environments, natural selection would have favored any genetic mutations that lead to a decrease in skin melanin content. Why? Because that's going to maximize your ability to make vitamin D. And that can be particularly important the further towards the poles you go, because there are some places where the total amount of UV that you receive is not enough to meet your body's vitamin D requirements. Okay. All right. So we've got these three hypotheses, really just two that are probably good ones. As you've probably figured out, these are not mutually exclusive, right? And in fact, most anthropologists today would argue that the global distribution of pigmentation that we see reflects a balance between these two ideas: the need for protection in places where UV is high from things like folic acid breakdown, and the need to maximize vitamin D potential in places where UV is low. Okay, right, there's our UV map again. All right, so whirlwind tour of genetic evidence for these different hypotheses. There's a lot of genes we could talk about. We're just going to talk about a few, but we're going to start with one of my favorites, which is a gene called the melanocortin-1 receptor, or MC1R. This is a great little gene, and I mean little. It is only about 1,000 base pairs in length. So the protein it produces is only 315, 317 amino acids long. It's very compact. In the days, now we use different DNA sequencing technologies, but back when this work was first being done, a short gene was very easy to sequence. It was just, it was great to work with, very, very easy to work with. So there's one reason why I like it, but the other reason why I like it has to do with what this gene does. So the protein that MC1R produces sits on the membrane of those melanosomes. And when your MC1R gene is functioning, I'm going to say properly, it's a little risky, but I'm going to say properly, it basically tells your body to make eumelanin, right? That brown, black melanin. However, in humans and in lots of other species, there are certain mutations in the MC1R gene that say, nope, we're not making eumelanin, we're flipping a switch and we're making pheomelanin. So in humans, MC1R is associated with lighter skin, but the very distinctive phenotype that it is really associated with is not just lighter skin, but fair skin, red hair, and freckles. So obviously this is my favorite gene, right? I feel very personally attached to it. So, but besides that, it's very interesting evolutionarily. So if we look at the protein of the gene, right? And so all these little circles here, these are the different amino acids produced by MC1R, and they're joined together in this protein that sits in the melanosomal membrane, right? That's what this green block is, and the protein sort of goes in and out of the membrane. And you'll notice on this diagram, right, there are some red and green and different colored arrows, and those are pointing out the locations of key mutations in the MC1R gene. Okay, and for a small gene, boy, there are a bunch. Okay, really, really unusual in that sense. And so in thinking about how does variation in this gene fit or not fit with those hypotheses that we talked about, well, we can start with the folic acid hypothesis first, right? And that hypothesis is basically going to say, hey, I think that if I look at MC1R sequence variation in people that are from populations that are primarily from these high UV regions, I don't think I'm going to see a lot of mutations, right? Because any mutation that flips that switch from eumelanin to pheomelanin, okay, and makes skin lighter, natural selection is probably going to have acted against those over time. And if I do see mutations, they're probably going to be a type of mutation known as a synonymous mutation. So a synonymous mutation is when the nucleotide changes, but the amino acid that's produced does not. So it's kind of like a sneaky, invisible mutation. Okay, and when researchers, many researchers have looked at this, and when they sequence that MC1R gene, yeah, they do see that mutations that affect phenotype, mutations that would flip the switch to pheomelanin production, they're generally pretty rare in these high UV populations. And so that fits our expectations that natural selection has acted against them. Now, when we look at MC1R sequence variation in populations from low UV regions, right, the vitamin D hypothesis would predict we should see lots of mutations, right? And they should all be the non-synonymous kind, the kind of mutations that change the amino acid and the protein that's produced. And here the evidence is a little bit equivocal. Okay, we do see plenty of mutations, particularly in populations from Northwestern Europe. We don't quite see enough for this to look like a really, really strong case of directional selection. So that this could be a case of maybe we see all these mutations in MC1R due to a more relaxed selection model. But definitely we see evidence for conservation in high UVR regions, and that fits what we would expect from that folic acid model. So the next gene we'll talk about is my other favorite gene, and it has the unfortunate name of solute ligand carrier 24A5. I don't name them. We just call this one SLC 24A5 for short. And so this gene is one of my favorites because it was one of the last genes I looked at as part of my dissertation work. And so this gene was brought to the attention of my graduate advisor, Dr. Mark Schreiber at Penn State University, by a colleague of his at Penn State Hershey Medical Center, Dr. Keith Chang. And Keith works with zebrafish. He uses zebrafish as models to understand complex human traits and human disease. And Keith called up and he said, hey, I've got this mutant zebrafish. It has a mutation in this gene, this SLC24A5 gene. And those mutant fish are a lot lighter. And you can see that in this image, right? The golden zebrafish is on the bottom, the wild type is on the top. And he said, what does this gene do in humans? And we said, we don't know. This is not one of the canonical 5 to 7 classic pigmentation candidate genes. We don't know what the heck this does. But at the time, we were very, very lucky. At the time, a new public database was being deployed known as the International HapMap Project. And the goal of the International HapMap Project was to type common mutations in a globally diverse, it was not globally diverse, a globally diverse set of humans, with the goal of trying to identify mutations that were associated with common traits or diseases. There was no phenotypic or disease information in this database, but if you wanted to know something about the frequency of a particular mutation in these populations, you could go in and look for free. And so that's exactly what we did. And we found that there was a one mutation in this gene, a non-synonymous mutation, so it changes the protein. Okay, and at this particular place in our genomes, right, you could have an A nucleotide or you could have a G nucleotide. Okay, and right away we noticed something very peculiar when we looked at the frequencies of these A and G alleles in the different HapMap populations. So there were 4 populations typed in the HapMap. There were individuals from Utah that were a stand-in for people from Europe. That makes sense. There were Han Chinese, Japanese individuals from Tokyo, and there were Yoruba individuals from Western Africa. And when we looked at the frequency of these alleles in the different populations, we saw something very unusual. And that was that every single person in that Utah European sample had the A allele. All of them were homozygous to it. So they had 2 copies of the A allele. Everyone else in all the other populations was homozygous for the other allele. That is not how human genetic variation works. It is very unusual to see an allele that is private to one population, right? Most genetic variation is shared across multiple groups. When we see patterns like this, one of the things it suggests to us is possibly this is something that's been affected by natural selection. And so we said, well, this is interesting. Let's not get too excited. Let's see if this actually affects phenotype. And so in the lab, we had a sample of African Caribbean and African American individuals that we were using in pigmentation studies. And we had melanin index values for those individuals. And in fact, we saw that, hey, if you have 1 or 2 copies of that A allele, your melanin index is much lighter than if you have 2 copies of the G allele, right? So the x-axis in this graph is actually showing a change in melanin index, delta MI. And so in fact, what we learned is that this mutation could explain about 30% of the variation in melanin index between people of European and African ancestry. That is a huge effect size for pigmentation genes, okay? We already knew about lots of other genes that affected pigmentation. None of them had an effect that big. So this was very, very unusual. I mentioned at the start, I'm really interested in evolutionary processes. I was very interested in the way natural selection might have affected this gene, right? Because it seems to fit very well with predictions from the vitamin D hypothesis. So I went and I typed this mutation in a bigger set of samples, about 1,000 individuals from 52 populations. So all of these pie charts are a population. And the yellow shading on the pie charts is the frequency of that allele associated with lighter skin. The turquoise shading is the frequency of the other allele. And again, you can see there's this really distinctive geographic pattern, okay, where we see the allele fixed at really high frequencies in Europe and mostly in Europe and Central Asia, and it's pretty rare everywhere else. And I'm not going to get into the statistics of we use, but obviously my question was, how rare is it? A pattern like this is like in like the top 1% most extreme allele patterns that you would see. So that was one really interesting thing. We said, okay, we're probably clearly looking at a case of selection. But the other thing that we noted was, we don't really see that allele in a lot of these Eastern Asian populations. And that was a surprise because the thinking at the time was that as soon as modern humans migrate out of Africa, depigmentation happens almost right away. And that would imply that the same alleles were involved in lighter skin in all populations living in lower UVR regions. That was what people thought. Instead, what this suggested to us is that, the genetic mutations that maybe are explaining lighter skin in other parts of the world are not the same ones that are explaining lighter skin in Europe. This is effectively a case of what we would call genetic convergence, okay? You might say, all right, well, this is one gene. Does every gene tell that story? Well, some don't, right? There are plenty that don't. But when we looked at 2 others, SLC45A2, very, very confusing naming, and the gene tyrosinase, we see a similar pattern, right? Not as extreme, but the same pattern is there. And so some of the takeaways from this is we could say, okay, well, very clearly natural selection has favored mutations associated with lighter melanin in these ancestral European populations, and that's very consistent with the vitamin D hypothesis. In many cases, the selection was recent and strong. So I was very interested in the timing of selection. I looked into this, and then some folks that are much better with the modeling did it. And in right, so I can't claim that work, but essentially it looks like selection drove some of these alleles to really high frequency in populations from Europe, maybe less than 15,000 years ago. Okay, modern humans have been walking around Europe 40 to 45,000 years. This suggests a very late and very, very rapid change in appearance. Not at all what we expected. And then the other flip side was we think we have genetic convergence going on. So the genetic architecture of lighter skin between European and East Asian populations is not shared. Okay, so you might be saying, oh, well, okay, next step seems easy. Just go use those same methods and find the genes that are important for lighter skin in Eastern Asian populations. Yeah, that was a lot harder. So I looked, lots of other people looked. We could not, using those same methods, find genes in the same way. And what that started to suggest was, it seems like the model in Europe is lighter skin has been driven by changes in a few genes, so maybe that 5 to 7 gene model, a few genes that have really big effect. But the situation in Asia might be very different. It might be, in fact, that in this case, we have many genes that have small many genes that have small to moderate effects that combined lead to this shift in phenotype. And those would be much harder to detect using the methods that we were using. So the way that people would look for that now is with a technique known as a genome-wide association study, or a GWAS study. And so the way that basically works is you get a big population sample, and it only works if you've got a big sample, and you measure them for whatever phenotype you're interested in. And then you basically genotype them for hundreds of thousands of mutations across the genome. And you're going to test for an association between every single one of those mutations and your phenotype. And because you're doing hundreds of thousands of statistical tests, you have to do all sorts of corrections. That's not the topic of this talk, but don't worry, right? They control for that. And when you look at those results, right, you might display them in a figure like this. So again, this is not a paper from my lab. This is a paper from Kazib Adhikari's work. He's now at the Open University in London. But GWAS results are typically displayed in a plot like this, known as a Manhattan plot, because it looks kind of like a city skyline. Okay, so along the x-axis, we have the different human chromosomes. The different dots that you see are p-values, right? The statistical significant values. Okay, so those are plotted on the y-axis, but they're plotted as the log of the p-values. So the higher the value, the higher the tower, the more significant the result. Okay, and so when you're looking at these plots, you're looking for really tall skyscrapers to tell you that you found a gene or a locus that's associated with your trait. And in this GWAS, they were looking at a couple traits, but they did identify 2 genes that might be involved in, that look like, not might, they are involved in lighter skin color in these East Asian populations. Okay, and the first is a gene known as OCA2. This is like a known suspect in pigmentation genetics, right? So mutations in OCA2 are associated with a type of albinism. Other mutations in and around OCA2 are associated with blue eye color. Other studies had suggested OCA2 might be involved in lighter skin in East Asians, but this kind of helped nail that down. So this was very exciting. And then they also found associations in a gene that we hadn't really heard of before called MFSD12 that also seem to be associated with lighter skin color. So really good preliminary work. There's a lot of follow-up that needs to be done. But the fact that they did, but one of the things that this suggests is that we really need to, I'm running out of time, so I'm not going to talk about this figure. Probably the model for pigmentation change and the genetic architecture in this part of the world is very different. Probably it's regulated by lots of those mutations that have moderate to small effects. We're not going to find an East Asian version of SLC24A5. Okay, so that's something we've got to keep in mind, that the situation is very complex. Okay, and to be sure, we've asked these same questions about other parts of the world: South Asia, Africa, Melanesia, again, which is where I got my start. And I have these images up here just to remind us all, because it's very easy to forget. in all of these places that we're talking about, pigmentation is really variable within these regions. Okay, so it's not correct to talk about like European pigmentation or Melanesian pigmentation or African pigmentation. There's a lot of diversity there. Most of the studies that we've talked about so far have been focused on big sort of interregional differences, but there can be a lot of diversity even within very small regions, and that can complicate our analyses and can complicate the models that we need to use. And so that's the direction that we're moving in. So thinking about sort of take-home lessons, right? Skin pigmentation is definitely a continuous, not a categorical trait. Global patterns of human skin pigmentation reflect sort of long-term adaptation to ultraviolet radiation. Pigmentation is a complex trait. It's under the control of lots of different genes, and those genes may have a complex evolutionary history that is not always the same from place to place. And so one of the things that we as pigmentation researchers need to start doing is not only using more diverse samples and thinking about sort of the local population history and the local context of those groups, but recognizing that sort of a one-size-fits-all model for pigmentation evolution probably won't work, right? So if we want to understand this better, we have to move away from that sort of heavily European model of few genes, big changes, and think about more complex patterns. And I know I talked for quite a while, so I'm going to stop there and take a breath and let Bronwyn tell me about all the questions that you all have. And thank you. Oh, and actually, while we're getting this up, I just want to put up an acknowledgement slide, recognizing my collaborators and funding sources over the years, as well as different students and technicians that have worked in my lab. Sorry.
All right. Thank you so much, Dr. Norton. Can we unshare?
Yes, let us stop sharing.
Okay, and we'll come back. Let me put a spotlight on you. Hold on. Let me figure that out. Okay, there you go. So you won't go right. Thank you so much. That was fascinating. It's time for questions. Do we have any questions? Richard asked, are there dominant recessive interactions among the genes?
Yeah, absolutely. So different mutations in all of these different genes, right? Some are going to act in a dominant manner. Some are going to act in a recessive manner. So the mutations in the MC1R gene, right, particularly the mutations that cause this phenotype, are recessive, right? So you need to inherit a copy from mom and dad to display the phenotype. Interestingly, there are so many mutations in that gene. You don't have to inherit 2 copies of the same recessive mutation. You can be what's known as a compound heterozygote and still display the phenotype. But Richard, you also mentioned interactions. And so you've kind of hit on like, oh, this is a mess. Okay. I did not touch this here because this is hard to model and we tend to ignore it. But yeah, guess what? The proteins of these genes interact with each other. Okay. So you could, for example, have 2 recessive MC1R mutations that should lead to lighter skin color. And the effects of those mutations are going to act with mutations at other pigmentation genes, such that you might not see a big reduction in skin color. So for example, I mentioned I did my graduate fieldwork in Melanesia. There's a much higher frequency than you would expect of a non-synonymous mutation in those Melanesian populations, right? We would think it should not be there. One of the reasons why we think it's there at the frequency that it is because other genes are the effects of other pigmentation genes are basically masking it, right? So they're still producing enough melanin to make sense.
All right, we have, let's see, what is the function of whatever protein SLC24A encodes?
So yeah, that's a great question. So so this protein is another melanosomal membrane protein, and to the best of our understanding, right? I don't think this has been updated. It probably it's basically an ion exchanger, so it affects melanosomal pH, and so it turns out melanin production is pH dependent. And so there are other genes like that OCA2 gene that also affect melanosomal pH. And we think that by affecting the pH, that affects the quantity of melanin that's produced. But yeah, that's a really good question.
Earlier on, Leah was asking if melanin is higher in higher altitudes, how do we explain that Swiss people in the mountains are white?
Yeah, so what I was saying was UV is higher at higher altitudes, but your question still stands, right? Because it seems like, well, if UV is so much stronger, shouldn't their skin be a little bit darker than it is? There are actually lots of populations that are not good fits, and sometimes there are explanations for that, right? So some of it might have to do with duration of occupation in that particular region, right? Like, so, I know in the European case, we talk about SLC changing pigmentation really quickly. There are other parts of the world where maybe we see these pigmentation mismatches, and it has to do more with like, well, people maybe just haven't lived there long enough for mutations that change phenotype to adapt. The other thing, right? The other thing I'll point out is like, as humans have evolved, right? Like I've been talking here about genes and biology, but like culture is really our jam, right? We've invented all sorts of ways, all sorts of things that help us survive and thrive in different environments, right? Um, you know, sunscreen today, but also like we wear clothes and hats and things that provide some protection. The other thing that I didn't talk about, and I'm not saying this is related to the Swiss, but another form of adaptation to UVR is tanning ability, right? So there are some people that tan really well. There are some people like myself where that's not going to happen. But tanning is thought to be protective, right? And it might be particularly important in places where you have really sharp seasonal swings in UV exposure. So I can't answer the question specifically about the Swiss. I'm kind of giving you just a general statement that these correlations aren't perfect, and there are always going to be populations that don't quite fit.
Now, Nick's question, I'm a little bit confused of, because it says, does scale pigmentation work in similar ways to skin pigmentation?
You're thinking about like fish scales? Fish or reptile? Okay.
Yeah, okay.
Well, I really like people, Nick. I am very much a people mammal type person. I can tell you, right? So scales are a little bit tricky, and I'm going to exhaust all of my knowledge of fish in 5 seconds. But so, for example, we know in those zebrafish that I was showing you, zebrafish don't have melanosomes; they have melanophores. It's an analogous structure. Fish scales are also tricky though, because they have that iridescent property. I don't know anything about that. I think like in general, principles are similar, but there are going to be some differences, and I'm just not educated enough about it to answer. Okay, good. You were going to ask about iridescence next. I know enough to know that that's important, but not enough to know exactly how it works. So I'm sorry about that.
Well, and that kind of piggybacks on Richard's, is that how does what you're studying, the mechanisms, are they the same in animal skin color? Is the same for humans or hominids?
So if by mechanisms you mean genetics, then yes, right? They should be similar, right? And for a long time, what we thought we knew about human pigmentation genetics was driven largely by mouse coat color studies, right? So yes, they are similar. If you're thinking about like how do other animals like mammals or chimps or gorillas, how are they making vitamin D? Are they worried about folic acid breakdown? I think yes, the general principles hold, but like there are right, there are other factors at play, right? We're a weird mammal in that we're naked, right? All of these other mammals are they've got fur and stuff. Oh, protein chemistry and stuff. Yeah, I think it should basically work the same. Again, that gets a little bit far out of my area, but my general thinking is yes.
And I think it's amazing that you said that the shift in the Central Europeans was in less than 15,000 years. I mean, that's quick.
Is lunacy. It's absolutely like when I first tried to do the math to figure that out, I was a postdoc at that time, and we just kept saying there's no way. Like, because what it implies, that type of rapid change implies that selection was very, very, very strong. And so the other, so to give you an example, the other sort of rapid phenotypic change that we see in European populations at about this time is the evolution of lactase persistence, right? The ability to digest milk sugar. Okay, and that's also a very, very strong selective force in human evolutionary history. And this the strength of selection here was estimated at one time to be kind of on par with that. So
Well,
yeah, so go ahead.
So sorry, any of the, are there any thoughts about the earliest hominids in terms of the skin pigmentation and
Yeah, that's a great question. And so you mentioned Nina Jablonski when we first came on. This is something that Nina has written about, right? So the the general thinking is that our hominid ancestors, right? So they're originating in Africa, right? Probably Eastern or Southern Africa, right? So that's clearly a UV intense environment, right? But also, right, they had fur, right? So then you get into these questions of like, well, what did they look like underneath their fur, right? And what happened when that fur was lost? Did that spur a big pigmentation shift? And so the thinking, I think, based on comparisons with other living primates, is that beneath the fur, pigmentation is lighter, right? Maybe not like light like me, but lighter, right? And so since that's what we see in a lot of these other primates, we're sort of assuming that would be kind of the ancestral state. But so then you wonder, well, what happens when we lose our fur? And the prediction is that when that happened, there would have been basically what's known as a strong selective sweep, okay, which is the same thing so at SLC, we saw a selective sweep for this light allele, probably at the time that fur is lost, which is associated with all sorts of other interesting changes like bipedalism and changes in thermoregulation. There was probably a selective sweep for genetic variants that led to an increase in melanin production, right? So it's actually really exciting to think about the changes that were happening in our lineage at that time, right? We're walking upright, we're losing our fur, skin's getting darker, we're probably starting to eat meat and make different tools. It was a busy time in our history. But yeah, and there's some evidence, for example, that at the MC1R gene, there was a sweep in that gene, maybe dating to around that time, that led to kind of the elimination of all alleles associated with lighter skin or pheomelanin production. So I mean, that's what the model says, right? We can't go back in time.
Yeah, well, and do the models take into consider? I guess they would have to take into consideration any of the climatic differences at the time.
Yeah, so we can use information about climate or climate reconstruction to try to think about, well, what makes sense at the time for these types of models where we're trying to predict, like, okay, what was this sequence like then? Those are more models based on like what we know about mutation rate and generation time and things like that. You can sort of use those models to figure out when all the modern day MC1R sequences would have coalesced to a common ancestor. But yeah, certainly you would do that, and then you would want to check and be like, well, does this make sense given what we know about where this hominin was living and what we think the environment was like, right? Because if it doesn't make sense with that, then maybe your model's wrong, or maybe you need to rethink something about the whole process.
LB has a question. How does interracial ethnic group DNA play a role in the melanin selection?
I'm not sure I totally understand the question, right? If the question is about like interpopulation, like population differences in melanin content and pigmentation genes, I mean, we're going to see variation from population to population in the frequencies of these alleles, and you know, if they're having an effect on pigmentation, we'll see that tied to the phenotype. But LB, I don't know if I totally understand the question. I don't know if you want to type a clarifier in there or not. Sorry. Good. Well, while LB's thinking about that, I'll just say, since you brought up our ancestors, there's a lot of, you know, I'm sure many of you are aware now, we're getting better and better at recovering ancient DNA from hominid remains, right? So we've sequenced Neanderthals, we've sequenced these mysterious creatures known as Denisovans, right? And so one of the fun things that people like to do is to use those DNA sequences to predict what those individuals look like. And I tend to caution very heavily against that. But for example, before Svante Pablo's group produced the full Neanderthals whole genome sequence, one of the genes that they targeted as a test case, just to see if it was possible, was the MC1R gene, right? Because remember, I told you it was it's so little and petite and easy to get at. They said, we'll try this. This will be an easy success. And interestingly, sequences of Neanderthal MC1R genes showed that they harbor mutations that lead to increased pheomelanin production, right? So consistent with the vitamin D hypothesis, but those mutations are not shared with modern humans. And so that's really cool, because that means that they underwent maybe a similar lightening process that modern humans underwent, but they did it their own way, right? So it's another case of convergence. But in general, I get a little wary of reconstructing phenotype in those fossils, because we can only look for the mutations that we know are segregating in human populations today, right? We don't know if there were other mutations segregating then that would have interacted with those and impacted phenotype. So I think it's always going to be imprecise to do that.
Yeah, it's interesting because if you go back and look at some of the fossil remains in the first reconstructions of different animals and how we've more that we've learned, and they go back and say, oh my God, I can't believe we thought it looked like this. It really looks like this, but we don't really know.
Yeah, I mean, some of those reconstructions have changed a lot as people have like paid attention to and been like, oh wait, this was a high UVR environment, like, we need to make the skin darker or whatever. But when you get to more recent stuff, like the reconstruction of Cheddar Man in England, those reconstructions showed Cheddar Man, who's relatively recent, as being very, very dark skinned, and that promoted all sorts of backlash because people didn't like that there was a Black British ancestor. But then and that reconstruction was actually based on the fact that the Cheddar Man DNA didn't have that SLC allele, right? But they went really, really far in the direction of making skin look really dark. And I don't know. I said, I don't know if you know all of that for sure. Now there's a lot of unknowns in that modeling that I think we need to be careful about. But it's fun, right? I don't deny, like, it's a fun activity.
Right. Any other questions? Okay, it's quiet. We are smarter. Everybody looks smarter on here than you did when you started, so that's good. And Heather, thank you so much for taking your time in isolation, in quarantine.
Less than 12 hours and I'm out of here. I'm very excited. But yeah, so if I appeared a little overly exuberant, it's just because I haven't been able to talk to people in a while. So thank you so much for letting me share this evening with you, and for asking such great questions. I really enjoyed it.
And everybody stay well, stay curious, stay outside, and go get your vitamin D.
That's right. Go outside and make some vitamin D tomorrow. That's what I'm going to do for sure.
Everybody take care. Take care. Thank you again.
Bye. Good night.
Good night.
Bronwyn, thank you so much.
No, thank you. That was wonderful.
Super, super. Well, enjoy the rest of your series and have a great spring, okay?
Okay, take care.
Thanks. Bye-bye.